Okay, guys, so last we were talking about media. We were looking at a blood agar plate. This blood agar plate had 5% sheep's blood in it, and we realized that organisms would grow differently on that plate. So we said it was a differential plate. We said that there would be different kinds of hemolysis going on. Well, now we're going to look at a phenylethyl agar plate. So PEA is what we'll say, right? PEA for phenylethyl agar plate. And this is similar in our discussion because this PEA plate actually does have sheep's blood in it. But I'd like to encourage you to ignore the fact that there's sheep's blood in this for now, which technically would make it enriched and differential. But let's just focus on the fact that there's phenylethyl alcohol in this particular plate. All right, as we go through these plates, I'd like you to pay attention to what is in the plate that makes it special. Okay, what are the key ingredients? Not everything that's in there, but what are the key ingredients that are in there? Okay, so what it is about phenylethyl alcohol that makes it special is that it actually really inhibits certain types of organisms from growing, and those are the gram-negative organisms. It is selective in that manner. All right. So we've talked about complex. We'd have like TSA, lots of different stuff to allow things to grow in general. Differential, where you would have a different way of growing. Different organisms would grow differently on a plate or a medium. And now we see selective. Selective means that it's going to kill certain types of organisms and select for others. Okay? So here we have, it's handy to recognize what is the plate going to look like before anything is put on it. So here's an uninoculated plate. In other words, the inoculum, the small sample of bacterium you would place on that plate to do a street plate maybe. There's nothing on it, right? So it starts out with this dark kind of red color, just like the blood agar plate. And if we were to place a gram-negative organism on this plate, do a streak plate on it, and you can kind of see this back and forth look, like it's been scraped a little bit with an instrument with a inoculating loop. Well, this has already been inoculated with an organism, and yet it looks very similar to the uninoculated version. That's because none of those cells stood a chance. These had to have been gram-negative organisms. As soon as you see that kind of a result where nothing grew, you know something about it, it's gram-negative. If we've done a gram stain, we better hope that that matches, right? That maybe they came out kind of a pinkish reddish color when we did a gram stain. This is a way of confirming that. Now, if we look below, we actually see a blood agar plate over on the left and a PEA plate over on the right. They look very similar, and they were inoculated with the same organism, which was a gram-positive cocci type of organism. Okay. Now, this PEA is selective for those gram-positive organisms, not against, right? And so they grew quite well. Doesn't seem to be much difference between the two plates because PEA didn't do anything to inhibit their growth. So we can say if there's no growth, like we see up here, gram-negative. If there is growth, like you see down here, we have a gram-positive organism. All right. Now, one kind of special plate that is PEA plate here on the right. Okay, so we have our PEA plate, and we have our blood agar plate, so BAP plate, over on the left. The same sample has been placed onto these two plates. The sample was a urine sample from a person that was having some signs and symptoms that indicated maybe a urinary tract infection. Now, on the right, you see these little pinpoint colonies, these little tiny pinpoint colonies everywhere here, right? Indicating that there's an organism that must be gram positive if it's growing on a PEA plate. But over on the left, we see not just those little pinpoint colonies, because they're still there, right? But we see these other bigger white, a little bit translucent, but kind of a white color colony, which is indicative of E. coli. All right? This person had a urinary tract infection, very likely caused by the E. coli that is growing on this blood agar plate. E. coli is a gram-negative organism, so this makes sense, right? The gram-negative organism can't grow on the PEA plate, but it is growing on the blood agar plate. This other organism, the Staphylococcus organism, is a normal organism. You should expect to find that in any urine sample. You find a certain amount, and it's not a bad thing, right? It's not something that would cause infection necessarily. Okay, the next plate that I'd like to talk about is a mannitol salt plate, a mannitol salt agar. Again, it's handy to know what a mannitol salt agar plate looks like before you do anything to it, right? So here we have an uninoculated plate over on the left. It looks kind of a little bit brighter red than we were seeing with blood agar and PEA plates. That blood in the other plates really makes it a little more opaque. It's harder to see through. But here you can see through it. And it's key to know, like we were saying, it's key to know what's in these plates. Okay, so what we find in a mannitol salt agar, and if you just kind of think a little bit, it tells you we have salt, high concentrations relatively with 7.5, mannitol, which is a type of alcohol that has sugar properties. It is an organisms can get energy from mannitol like they can from sugar, if they have the enzymatic capability, of course, right? And the other thing we see in this plate is a phenol red pH indicator. Now, phenol red is going to be red and even a pink color when it's basic. Well, I'll say a pink color when it's basic, a red color when it's more neutral. So what we see here is a relatively red color in a more neutral environment, okay? So what we're looking for here is, will that neutral environment change as a result of growth? A lot of organisms, when they utilize a sugar source, or in this case mannitol, which is like a sugar, they will produce acidic byproducts. And this will change everything to a lower pH, which would cause phenol red to change from a red color to a yellow color. So if we look at this plate here on the top right, I see some etchings here, right? These etchings indicate to me that this has been inoculated and there's no growth. This was in an incubator for 24 hours at 37 degrees and nothing happened. This would be indicating to me that it cannot grow in this environment with all that salt. The salt is giving this plate a selective characteristic. It will select against organisms that can't grow in the presence of salt. Halo is going to be our salt prefix, and phile is lover, right? So this is a non-salt lover, non-halophile. We know it because it didn't grow. And a whole slew of organisms that we work with will not grow on this plate. What we're usually looking for, if we are interested in using this plate, are a lot of times halophiles, in particular the Staphylococcus species, happen to be halophiles a lot of times. All right, so let's look at the bottom two then. Down at the bottom we see some streak plates, some good ones, because I see some nice isolated colonies here, right? Somebody did a good job, and I see some down here on the bottom right as well. And I see that there is growth, first of all, which tells me they are definitely halophiles. They have no trouble growing there. They are not selected against. They're selected for, right? This is part of the selective characteristic of this plate. But I do see a definite color difference, okay? With this bottom right plate, the one thing I don't see is a color change to yellow, which is what we're looking for. If there are acids produced as a result of growth, I would expect to see a yellow color. And I see no yellow down here at all. This tells me that it is capable of surviving the salt. It's a halophile, but it is a non-mannitol fermenter. It does not break down mannitol as an energy source. And it does not produce acidic byproducts. So we don't see a color change down in here, okay? But we do see this color change down the bottom left, telling me that this is a halophile and a mannitol fermenter, okay? So in this case, the phenol red's the pH indicator, and it's going to turn from the red to yellow if there's acidic byproducts showing us that there is a mannitol fermenter there. So here we have another plate. I kind of like this plate. I just want to use this as an example, as a little bit of a review. So what they've done here, I'm just going to split up the plate. On both of these plates, they have split up the plate in kind of into thirds. Okay? So A, B, and C are the different species. A and B clearly grow. Up here we have a TSA plate. So every organism grows. There's nothing there to kill any of these organisms. But on the bottom we have, in the C third of this plate, we have absolutely nothing. Nothing at all, right? This indicates that there was no growth. So, mannitol salt agar, no growth means non-halophile. Okay? Why did it grow up here? Well, there's nothing there to kill it, right? There was no salt in that plate, or very little salt. Now up in the A and B third of this plate down below, we do have growth, which tells me that they're both going to be halophiles, but we only see a yellow color change down in here, right? So all of this yellow we see down in this third tell us that this is a mannitol fermenter. Now, before we call this clip quits here, I want to just point out that just because item C, this bacterium labeled C, was not able to, did not show a color change down here, does not mean that it's a non-mannitol fermenter. We didn't give it a chance, did we? The salt killed it. So we cannot pass judgment on a non-halophile within the mannitol salt agar. We can't say for sure that this organism C is unable to break down mannitol. All right, so we just, that's just one thing this plate can't give us. It can't tell us whether it can do that or not. Okay. And a little analogy I always use for this is, you take me and you drop me in the middle of the Mojave Desert, right? And you may look for evidence that I was there, right? Now, if I can't survive in the Mojave Desert for more than a day or two, I will not be producing waste, right? I mean, this is a corny way of talking about it, right? But really, it's the waste products of these organisms that cause the color change, right? So here we're doing that to bacterium labeled C. This organism was not given a chance to survive. It was not given a chance to ferment mannitol, so we can't really pass that judgment. Okay, so let's call it quits. We'll move into the next few plates here in a sec.